Dr. Carter is a computational geneticist focused on the biology of several complex traits, and today is going to be talking to us about his work in mouse models of Alzheimer's disease. Dr. Carter actually got his PhD in physics with Paul Ellis at the University of Minnesota in 1997 and has pretty much been hanging around the 45th parallel ever since. He's done postdocs at the Niels Bohr Institute in Copenhagen, Stony Brook in New York, University of Washington, and Seattle. It's during his time in Seattle that he made the transition from physics to systems biology, which he has told me was primarily driven by the large amount of data coming from the Human Genome Project and related projects. And then there he joined, in Seattle, joined the Institute for Systems Biology, where he was until 2010. Then he moved to further down the 45th to Bar Harbor, Maine, where he's currently an associate professor in the Bernard and Louisa Milch Endowed Chair at the Jackson Laboratory. He's also the head of bioinformatics for two National Institute of Aging funded centers addressing Alzheimer's disease at JAX by integrating genomics and molecular biology with model systems. He's also part of other related R01 and U01 awards. He's been a productive scientist, great collaborator throughout his career. I won't list his many contributions to science, but as I said, he's been particularly productive, particularly in the last few years. And I'm sure Dr. Carter will be telling us a lot about his work today in his talk titled Modeling Alzheimer's Disease with Cross-Species Genomics. Dr. Carter, take it away.
Thank you very much. And thanks for everyone's patience with the audio. Hopefully I can be heard now. And thanks very much for the invitation. This is really exciting. Been looking forward to spreading the word about what we've been doing in Alzheimer's disease, and the more people we talk to, of course, the more broadly we might have an effect on not only Alzheimer's research, but more broadly neuroscience and related disorders. So I'm going to talk about how we're modeling Alzheimer's using cross-species genomics. There will be no behavior in this talk. We haven't done a lot, and I'm not an expert on that, but a lot of neurogenetics. And so, yeah, so I'm at the Jackson Lab. I'll note that this theme around hovering around the 45th parallel, my lab is split across Farmington, Connecticut and Bar Harbor, Maine, the two JAX research campuses. So yeah, we're continuing to hit that latitude. So I want to start with where we really began this project, and it's the genetics of Alzheimer's disease. So like most other complex diseases, there are very large meta-analysis of Alzheimer's disease for genome-wide associations. And here is, a recent meta-analysis from, I don't know, it's probably something like 800,000 individuals, phenotypes and proxy phenotypes for family cases of control in UK Biobank. You put it all together and you get about, 40 to 50 significant loci above that magic 5 times 10 to the -8 level that the GWAS tends to favor. And it's really a catalog of the potential genetic factors driving Alzheimer's. I will say when people first started doing this in Alzheimer's, it came as a bit of a surprise to some researchers to see the very broad range of biology being represented by these candidate genes at these loci. So, we knew about ApoE4, a cholesterol transporter, not narrowly focused on neurons or neurodegeneration, was playing a role. And I think the lesson since then has been that same thing over and over again, that we're seeing very broad biology, immune biology, metabolic biology, et cetera, along with some, synaptic markers and that sort of thing too. But it's a real mix. And so it's been a real challenge to try to understand what the GWAS are telling us, and even more so a challenge to use that information to better understand the disease and how to treat the disease. And it really comes down to the fact that all of this is just statistical association between a known genetic variant that is either marked, found with full genome sequence or imputed, that is leading to increased disease risk. And how do we actually translate that information in a useful way? And so what my lab's been focused on, and what the project I'll be talking about is really built around, is trying to use multi-omic approaches to determine how variants affect traits. And so by multi-omics, I think we're all pretty familiar with these approaches now, but we have transcriptomics, measuring mRNA, proteomics, measuring abundance of proteins in samples using mass spec usually, and metabolomics, usually same technique, but a little different flavor, given that we're looking at metabolites there that cannot be directly linked to the genome. They're more functionally linked through their production or interaction with cellular components like proteins and transcripts. Okay, so using these three in combination, we're trying to better understand the disease in mouse models. Most of what I'll talk about here is transcriptomes, because that's where we started and that's what we have the most data on, but I will show some proteomic and metabolomic data and suggest where we're going there as well. So when we think about the disease and we think about using these multi-omic approaches, everything really starts with the human data, right? That's how we define the disease, is individuals who are diagnosed. And so when we're coming at this from the mouse side, that's what we want to use to guide our work. And so there's a big project called the Accelerating Medicines Partnership for Alzheimer's Disease, or AMP-AD, which is an NIA partnership with industry to better understand the molecular properties of Alzheimer's disease. And from that has come these three clinical cohorts of postmortem brain samples from ROSMAP, which is Religious Order Study and Memory Aging Project, which is run at Rush University in Chicago. They have submitted 700 samples to date from dorsolateral prefrontal cortex, both cases and controls, as well as a number of mild cognitive impairment diagnoses. The Mount Sinai Brain Bank had 300 samples, where they've in many of those individuals looked at multiple regions. 4 different regions are represented in their data set, shown here. And finally, the Mayo Clinic, where they have gone through and their study design was to take 270, look at cerebellum and temporal cortex, and really maximize neuropathology. So they're sort of selecting for high levels of neuropathology in the Mayo data set. And that's in contrast with ROSMAP, which is really more of a, it's a longitudinal study. It's an aging study, and so they're not particularly selecting in any way, and they're getting maybe a broader representation of neuropathology, a lot more mild neuropathology in those individuals, but diagnosed with Alzheimer's in many cases nonetheless. So we started with this set of data. These data are growing. We're about to double the size of this database. But this is where we began. And we worked with a group at Sage Bionetworks, one of the contributors to AMP-AD, to harmonize all these data. And that involved sample swaps across these sites to make sure that we could normalize across all of the different sites and different data sets, and have a sense of a global view. So it's, it's not a perfect harmonization. We can't directly compare the results of one study to another, at the very detailed level, because using different read depths and technologies. But it allows us to sort of put everything in the same level, sort of same playing field. So when we do talk about changes in one region of the brain, it's roughly as reliable as changes in another region in another cohort. Okay. So that took a lot of work, but you can, of course, access all these data individually. They had a number of publications, came out a few years ago. Here they are all on scientific data. All the data are also available on the AD Knowledge Portal, which is a centralized data dissemination center for not only AMP-AD, but what I'll talk about as well. So I think this is probably familiar to many people in this group, but I want to make sure we're all on the same page of what you do with a big transcriptome from a large cohort like this. And we could go through and look at cases versus controls and look at differential expression, and we would select from that sort of analysis maybe some key genes that are markers. But what has been found, I think, through a lot of work in both model organisms and in human data, is that if you think about these in terms of gene networks and think about perturbed networks across many genes that correspond with particular processes or pathways, that is a more fruitful way to explore and explain the data. And so when you have a population like that you don't want to start with partitioning subgroups like treatment versus control or mutant versus wild type like we all have in our model studies, and you want to think more about variation across a heterogeneous population, this sort of a network analysis is often a place to start. And it's usually fairly straightforward in that, you start with what we have on the left there is basically a big data matrix where you have genes expressed, usually, thousands of genes across a number of individuals. And usually, you can sort these data sets such that you can see sort of these blue and red blocks, suggesting that there are groups of genes that are changing similarly in groups of individuals. So there's a structure there. And so to extract that structure, we often use correlation analysis. So you can build a big, say, correlation matrix across all individuals for each pair of genes. And then you tend to look at that matrix and find the structure in it. And that can be represented on the right there by a network showing dots, which are the genes, connected by high correlation, which are the links. And then you use some sort of a community algorithm to identify structures within this network, and we tend to call those gene modules. And so in this toy example, those green nodes that are colored gray are the set of genes that are co-expressed together across the population, and that's a module. So module is a set of genes in this context. This can get pretty complicated, and I want to highlight the tool that many of us use as weighted gene correlation network analysis, WGCNA. It actually takes that correlation network and looks at correlations of correlations, so to speak. So finding genes with similar patterns across the entire genome in their correlation of other genes. I call this a topological overlap matrix. Here's the link where you can get to this package. There are other ways to do this. This is one that I think has really been the workhorse, and we use it in a lot of our work. And it was used in this human data analysis. What you get from gene network analysis after you do all this is basically this correlation matrix or topological overlap matrix in the case of WGCNA, where you can see these clear neighborhoods, these blocks where you have groups of genes that are co-expressed. WGCNA assigns them a color. That's how they name their modules. Gray is what's left over in the background, and the genes that get assigned a color or a module can then be displayed, say, in a network view. And so this is a real view of the toy one I just showed, where you can see there are a number of modules. Some of them are fairly close to one another, like the light blue and the green there. They're close to one another in the matrix. They're close to one another in that network. But the hierarchical clustering has called those as two distinct modules, so they're distinct but related. Okay, so this gives us basically a way to summarize, dimensionally reduce all of that data into groups of genes that are covariate and sort of you can think of as a unit within themselves in the data set. So we did this for Alzheimer's disease in those data I was just talking about. And so this is what we got. So this is a summary view of 30 modules. Each module has a color assigned to it because that's how WGCNA names the modules, but also a brain region identifying the study and sample region from which it came. So TCX blue is temporal cortex blue, that's from the Mayo study, and all the way down the list. Why 30 modules? Well, so there are actually some 250 or so modules came out of this analysis, but then we went through each module and asked whether that module was enriched in genes that were differentially expressed in cases versus controls. So in other words, these are the modules that we could then link to Alzheimer's disease incidence within the population. Okay, so these, we think of these basically as the catalog of genomic signatures that are mapping different aspects of Alzheimer's disease. One thing you'll note is that there's a block structure to the similarity matrix, which we're showing here. So the red blocks are basically modules with a very, very high overlap in gene content. So same genes showing up in different modules from the different data sets and different brain regions, suggesting that there's some sort of universal consistency in many of these regions associated with Alzheimer's disease. And when we look within those modules, see how the genes are annotated to different processes and cell types, we can really identify these, what we call consensus clusters or supermodules, where we have, a group of 3 modules that are very strongly enriched in astrocyte signature for the first 3. The next is really an inflammatory signature. Next is really centered on neurons. So that's, representing the loss of neurons in these samples, so reduction in neuronal gene expression. But we're also picking up things like oligodendrocytic signatures in cluster D across all the regions. And then finally, there's a sort of basic cellular processes set in cluster E, which has a lot of protein folding, heat shock response, other sorts of basic cell processes, DNA metabolism, DNA DNA repair, et cetera, that generally show up across most of the modules and most of the cohorts, but not universally. It's a little more scattered. And so I'm taking a minute to highlight these diversely represented processes here, because we'll come back to these a lot as sort of, again, our catalog of what's going on Alzheimer's disease and the sort of signals we would want to try to model. Some of these modules are really pretty simple. So here's TCX blue module. It's almost all astrocyte genes, as you can see on the right. It's almost all increased expression, as you can see on the left. So that's an astrocytic signature that is upregulated, and that's in cases versus controls. This is just showing the females, but you'll see very similar effect in the males, and correlated across all the individuals in the population. Some modules are not simple. So here's another one, CD turquoise, where you're seeing some genes go up, some genes go down on the left. And on the right, you can see that you get these clusters of different annotations to different cell types. So microglia, endothelial astrocytes. You can see there that this is a signature with increased endothelial and microglial gene expression and reduced astrocytic gene expression. These are all together in the same module because these three effects are all correlated across the population. Okay, so the modules can encompass a lot of diverse biology, but it's biology that sort of seems to be operating together in these data sets across these populations. Okay, so we're thinking about this as a way to go back to this problem of the genetics. How do we get from genome-wide association to disease risk? Can we use these modules to sort of interpret effects we might see from genetic variants towards disease risk? And of course, this is a very difficult thing to do in the human studies, because the big case control studies are powered by massive cohorts of hundreds of thousands of people. The genomics, the transcriptomes are 1,000, 1,500 samples over about 800 people. So they're not nearly powered enough genetically to do this sort of GWAS analysis of the modules. You can do that, and you do see some associations here and there, but you're not mapping effectively, with any clarity, these genetic variants onto specific modules. Doesn't have to be that way. It could have come out nice and clean, but that's apparently not how Alzheimer's works. Okay, so this is where model systems come in, and this is where what we call the Model AD consortium came in. So the idea here is that we want to try to capture all of this GWAS signal and all of the biology it's pointing to in mouse models and in new mouse models. So here I'm showing a cartoon of, say, the Alzheimer's representation in the human population, which is on the left there. About 5% are early onset cases where there's an autosomal dominant mutation, often in amyloid precursor protein or presenilins, that will cause an early onset disease. That's fairly rare in human population. What's far more common is late onset, age-related, or what used to be called, and sometimes still is, sporadic Alzheimer's disease. The existing mouse models are really built around those early onset mutations, usually transgenes with multiple of those mutations, driving a lot of, in most cases, amyloid or tau pathology. And those have proven to be very limited in terms of preclinical translatability. So in other words, well, frankly, we can cure quote unquote Alzheimer's about 400 different ways in those existing models, none of which is translated to a clinical success. Okay, so one way to tackle this problem is to expand our model base and start to come up with new models, maybe based more on the late onset genetics and more faithful to the biology of the late onset genetics, and use those then as tools to study the biology and also tools for preclinical analysis. And so this idea spawned the Model AD consortium. You can see we're model organism development and late evaluation for late onset Alzheimer's disease, to create and characterize these new models. And we have basically a commitment to distributing all of the data and all of the models without any restrictions. So pharma can use them, researchers can use them, you can take them and cross them with anything you want. It's a completely open approach to this problem. And that's our hope, is that others will see these models and go to work on them, because we have fairly limited bandwidth to actually phenotype them, despite the fact that we're a big consortium center. The main contributors are in the lower right there. Of course, Jax is involved, and me. We've teamed up with Indiana University and the University of Pittsburgh. For one center, the University of California, Irvine leads another center within this consortium, and Sage Bionetworks is involved in both centers in terms of data dissemination. So what's our strategy? So in our center, we're really trying to model the complex human disease from GWAS, like I was suggesting. So we're starting with the GWAS, we're introducing those genetics into the mouse models. I'll tell you how we do that. We're then taking those models, phenotyping them in multiple ways, omics, doing neuropath and imaging as well. I'll talk mostly about the omics today, but we're guiding all of that phenotyping around what we can align with human data. So we know there's human omics out there. I showed you that. There's human neuropath for many of these samples out there as well. And of course, in vivo imaging, PET and MRI are quite common in Alzheimer's research these days as well, and hopefully becoming more clinically relevant. So we're matching that. And so we're really structured, you see, around human genetics and human phenotyping to understand the relevance. Once we have relevant models that we think are some expression of Alzheimer's disease, we can then take those to a preclinical testing core, where the idea then is to target whatever molecular pathology we're seeing expressed in those models with a therapeutic that is going after that sort of pathology. So for instance, if we have a strong inflammatory signature in a certain mouse that matches that inflammation we see in human, is there an anti-inflammatory that might be proposed for Alzheimer's disease that we can then treat the mouse and see if we're engaging that. And this is, this is sort of a long-term idea. We've got to get the mice and move them, through this process and then match them up with a candidate therapeutic. But I will show you, a proof of concept of this sort of analysis by the end. So we start with the translational genetics. So this has really been a comprehensive view of the genetics, human and human GWAS results for Alzheimer's disease. Here again, I'm showing this latest meta-analysis, but we're casting as wide in as we can, looking at all GWAS publications involving not only case control, but relevant imaging markers, behavioral, cognitive decline markers, as much as those are used for GWAS. Those tend to be more difficult to harmonize across multiple data sets. So the big meta-analysis is not so strong there. But to the extent that people are trying to do that, and there are people putting a lot of effort into it, we're bringing that data in as well. And it also involves, pulling data from dbGaP and reanalyzing some of these data. And the reason is we want to get to variants. So we're really committed to trying to translate candidate variants into the mouse. And again, we want to do this to drive the disease, the real complex disease. But it's also, as a geneticist, I'm really excited about the idea that we just want to functionalize these variants. And at some point, if we really want to understand these GWAS hits, that's what we need to do. And so some of this you can do in cell lines. Many people are doing it. I think in some cases, an in vivo model is really going to be much more helpful, especially with complex diseases like Alzheimer's. And so we're giving it a go. We've got a center set up where our job is to make these resources. We can do it at some scale. And so that's what we're doing. And so we assess variants through many different criteria you can see there, and I sort of represented on the right how we sort of find those loci, and we're doing this for all loci. Just picked out a few here, asking if it's for sort of is our coding variant or non-coding variant candidates in functional regions. What would the predicted function be? And we have things shown there: variant effect predictor, roadmap, polyphen, CAD, etc. All those scores for both coding and non-coding variants. To try to predict whether there is a relevant function. Of course, we want to make sure that the variant is pathogenic, and that's sort of a, dependent upon the mouse reference and the human reference, such that if we edit the mouse, we're going to drive towards a higher odds ratio that you see in humans. We do like conserved variants, so we use GRP and DeepC. DeepC is a non-coding analyzer from Olga Troyanskaya's group at Princeton and Flatiron. That's not entirely necessary. We're starting to humanize more loci now, but you always worry a bit about compatibility once you humanize a gene. And so we prioritize conserved variants that we can go after. Of course, then we can CRISPR right in, which is what we're doing. We want that variant to be replicated or well represented in meta-analysis. And finally, we have a bias toward common variants. We have gone after some, 1% variants like TREM2R47H, very well known in Alzheimer's risk analysis. But for the most part, we're trying to capture common risk for the common disease. And so if we can find things at a high major or minor allele frequency, we'll bump those up our list. And so you can see how in many cases we have good candidates that make it all the way through this, this assessment criteria, and that's where we make our mice. A couple of examples. Here's a coding variant in the gene Pilra. It actually contains multiple coding variants. There are multiple good candidate genes in this locus too. However, there's a particular SNP that causes a G78R amino acid switch, and that allele has been shown to protect against herpes simplex virus 1 infection, and potentially through that mechanism reduce Alzheimer's risk. So there's a lot of work, especially recently, on the contributions of viral infection to susceptibility to Alzheimer's disease. And so, this is one of the examples of one of our coding variants where it has a functional change which we might be relevant through a particular mechanism, and here it's through viral infection. And so this is a good example where we created this variant, and we're actually working in collaboration with someone who's really interested in analyzing the viral role in Alzheimer's risk. And so we're not doing a lot to phenotype this mouse. That mouse has been shipped, and they're exposing the virus and then doing the Alzheimer's related phenotyping after that. I'm highlighting this because this is the way we really like to partner with people and try to spread what we're creating in terms of model resources and use them to address really specific questions investigators may have. We're also looking at non-coding variants. So here's BIN1. So BIN1 is the main candidate at this locus that is the second strongest associated locus in Alzheimer's disease after ApoE4. So it's the second tallest tower in the Manhattan plot. It's been there for a long time in terms of the genetics. It has no common non-synonymous coding variants. So it's been a bit of a mystery as what's driving this locus. Not surprisingly, most people think it's non-coding variation. That's probably what's driving quite a few of these genetic associations. And it's been historically rather difficult to prioritize these and interpret these, but that I think has really changed. Even since we started this project a few years ago, there are a lot of resources, a lot of annotations of the genome now where we can start to look at variants like these non-coding ones and start to ask questions. So for instance, we have the SNP. This is the one that, of course, will be the punchline, but how did we get to it? Well, so first of all, we went to Regulome-DB. Regulome-DB will combine a lot of existing data on genome annotations, especially non-coding genome annotations. You put in a region, you put in a SNP, it'll score it, it'll rank it, and it'll give you things like allele frequencies across a lot of different populations or databases, as you can see there. So this is a SNP that has fairly high representation, 30 to 50%, depending on which study cohort you're looking at. So common variant in a really strongly associated gene. It has a pretty good rank. So here's the ranking system that Regulome-DB uses. So this means that the SNP sits in a transcription factor binding site that is in a known motif. It's in an open chromatin footprint. And right in the middle of that peak. So not perfect because there's no EQTL association, but it's got a lot of good information. So within this region, this is the best one. In fact, we dug into the AMP-AD data a little more, where we also have whole genome sequencing, and actually saw that there is an EQTL. And so beyond what's in Regulome-DB, we looked in GTEx. You can see there's a whole blood association, but even more interestingly, in ROS-map brains, we see an association with the variant that it leads to reduced expression, which is opposite of what you see in blood. Okay, so this will be a theme that depending on which tissue you're looking at, you'll often see different genetic effects, and yet the effect exists in both tissues. So it's probably strong evidence that something's going on, but how you would interpret what's going on from the blood maybe is not obvious as something different may be happening in this case. Abundance levels of transcript may be different in the brain. Okay. And so this is how we get at some of these GWAS loci that are non-coding, is to look for annotations and then dig into some of this more specific Alzheimer's related brain data and see if we can buttress with EQTL or PQTL or whatever else might be there. So this is the SNP we're going after in BIN1. And I will note that if this data had been in Regulome-DB, it'd have a very, very high score, it'd be a 1B. So almost certain to perturb gene expression and hopefully then disease relevance. I will note that there is a big trade-off here between allele frequency and effect size, and everybody who struggles with GWAS and human genetics these days knows this is the case. We often get questions about, why do you go after a variant that has a 2% effect on disease risk? The simple answer is that's most of them. But the more complex answer is we're trying to dissect the biology behind those variants. So even though it may have a fairly small effect when you average over all the heterogeneity in a human population, when you dig into, say, omic signatures, you might see which biology you're perturbing there, and that may tell you something about the disease. And so we can go all the way from those early onset causal rare variants up in the upper left corner, down to the GWAS world of ApoE4, which if you're a homozygote will increase your risk tenfold, if you're a heterozygote twofold. TREM2 is one I mentioned, fairly rare, but also fairly powerful in terms of its risk. And then in the lower right corner, we have the big pile of all of the GWAS variants, high allele frequencies, very low risk profile. And that's really where we're digging now that we've more or less exhausted the others. I will have one little aside here that we're somewhat blind in that our populations differ, and we haven't looked in too many different human populations for these effects. And ApoE4 is a very nice way to illustrate this. If you look at what we have here, what I've mapped in terms of ApoE4 disease effect and allele frequency, it's really based. ApoE4 is typed by 2 SNPs. There are potentially 4 ApoE genotypes if you're using this coding. 1 you never see in the human population, 2 is protective, 3 is the most common reference, 4 is a risk allele. What I'm mapping here is from European-derived populations. If you start to look outside European-derived populations, it moves around a little bit. So in East Asian, in particular Japanese association study, you see a little lower allele frequencies and a stronger effect. So maybe even more relevant if you were to start from that population. But if you look within Hispanic-derived populations in the United States, suddenly that effect gets a lot weaker. And when you go to African-American populations in the US, the effect actually gets rather small. As a heterozygote ApoE4 carrier, the effect is really down in the GWAS range. So a minor risk increase, a higher allele frequency. And so all of this, of course, also, when we think about effect size of a GWAS variant, needs to be taken in a context from where we extracted the data. So again, maybe the effect we're seeing isn't the full effect that one might see on the biology when you move into a model organism. The interesting thing I will note here is this is not a genetic background effect, as far as we can tell. There was a nice publication suggesting that the African-American dominant E4 allele is different than the European dominant ApoE4 allele, not in terms of those typing SNPs I'm showing in the upper right, but in terms of non-coding SNPs in the area. And so there's a particular non-coding SNP that has been mapped, shows up very strongly in regular MDB that may be mediating that risk. And so this, and another thing we need to think about in doing this modeling is that even when we have a really strong coding SNP, it's a great candidate, the regulation is still relevant in the area. Okay, so we've made a lot of models using this translational genetics approach. And so can we go back to this idea and translate the effect of that genetics through, say, a transcriptomic or other omic output? So before I dig into that, I just want to note we've made a lot of mice and show you the slide of all the variants we've made. I'm not going to linger long enough so that you can read them all, but please look in the lower right corner. We have a strain table that keeps it updated, has details on all these variants. We're very interested in people getting their hands on these mice. So even if you're not studying Alzheimer's, some of these might be relevant to what you're looking at. MTHFR, for instance, is a vascular risk factor that we're trying to get that community excited about. PCALM is a gene that we have perturbed here in an AD context, also has been associated with other neurological disorders and conditions. And so please visit our strain table and see if there's anything of interest. Okay, back to this idea that we need to align what we see in these mice with human data. What do we see in these mice? Well, we do a lot of omics on these mice to begin with. So we go back to these gene modules where we have modules of coherent functions across multiple data sets and ask, do we see the same catalog of transcriptome changes in any of our model mice Or alternatively, if we have a model mouse based upon a particular genetic variant, can we identify which of these processes we might be perturbing that's disease relevant? Okay, so we don't have to do WGCNA and clustering because we have a very particular study design here. So I'm just showing this is mostly what I show for the the human clinical focus researchers to stress that when we do the experimental analysis in a mouse system, we have very structured, well-represented populations. We are well powered to look at both sexes. We are powered to look across different ages. Here I'm showing 4, 8, and 14 months, which is pretty much our standard. And then of course we have a number of genetic variants we introduce, sometimes in combinations. We can map all of this out in our sample maps and then analyze the data accordingly. So one way we analyze this data is to just do a linear model where we take the log expression of every gene, identify baseline expression, sex effect, age effect, and then the effect of whatever genetic variants, whatever knock-ins are carried by that mouse model. And again, in many cases, we have combinations of multiple models. And so from this, we can infer the effect of every one of these factors, sex, age, genetics, on the expression of each gene. And then we can ask, does that expression match human data? And the way we're doing that is to go to those human modules. And this is really driven by the idea that the modules, again, are the catalog, and we want to ask what we're perturbing within that catalog. So modules generally have a few hundred genes in them, and so we can ask whether the effects of a mouse factor in that linear model match the effects of disease in the human data. So that's what I'm showing here in these two correlation plots. On the left, you can see the x-axis is log fold change in cases versus controls of the genes within the SGG-Troyes module. And so every one of those red dots is a different gene. Some genes are higher, some are lower in Alzheimer's. And we're comparing that with the effect of a TREM2R47H mutation in a mouse, which drives some genes higher, some genes lower. Those changes are correlated with what you see across the human genes within that module. Okay, so here's a case where we'd say we have a significant correlation between the mouse variant perturbing the module in the same way that it is perturbed in Alzheimer's disease. On the right, of course, is a negative result where all of those genes are upregulated for the most part within that module in the human data on the x-axis, but say for ApoE4, we're not seeing that consistent effect. We see some up and some down, but no correlation. So whatever's going on there, might even be interesting for an ApoE researcher, but it's not mappable to what we see in Alzheimer's disease. Okay, so you can imagine we could do this over and over again. Every genetic variant we put into the mouse, every one of those 30 modules in humans, and you get a big matrix of these correlations, basically, asking if we perturbed each of these modules. And you start to generate charts that look like this. So here's one genetic variant, ABCA7, A1527G. It's a knock-in coding variant. And we looked at it at 3 different ages, 4, 8, and 12 months. And so that's what the rows are. Every column is one of those different human modules, and they're sort of summarized here by their class. So they're extracellular matrix modules, the first 3, immune modules, the next 7, neuronal, et cetera. And so we can just go through and compute that correlation in every one of these cases. And if there's a positive correlation, we give it a blue dot. If it's a negative correlation, we give it a red dot. So blue dots here mean we're perturbing that module in an Alzheimer's-like way. Red dots means we're perturbing the same module, but it's going in the opposite direction. It's sort of protective. And so we see these really interesting patterns. And here's a great example of ABCA7, where we're hitting a number of modules, but the effect is age dependent. That in the 4-month mouse, we have a few dots there, not a lot going on. In 8-month, we start to hit those oligodendrocytic and proteostasis, metabolism, et cetera modules, the mixed modules, pretty strongly, but they're all sort of in a protective direction. But as we age that mouse out to 12 months, things switch. So we've perturbed those systems in some way. That's how we interpret it, such that with aging, things actually start to look more Alzheimer's-like. And so that's what's shown by those blue dots. You can dig into the modules and ask, okay, what are the genes that are really changing there and what's their function? That particular module where we're hitting a very strong positive correlation, there's a lot of protein folding genes in it that are going in and driving that strong correlation. So there's an interpretation for what's happening with aging in the ABCA7 mouse. You can do this for MTHFR. Again, red dots sort of morphing into blue dots, sometimes going into red dots with age. So sometimes we're not hitting the pathways in an Alzheimer's relevant way. Other times we are. Again, we're hitting those last superclass of modules, those sort of general cell function. The module we're hitting the strongest here is really fatty acid metabolism genes driving that. So again, age-related genetic effects probably make sense for age-related disease and the variants associated with them. And then finally, some cases we don't see much changes with aging, maybe a little increase in correlation with aging as we're showing here for an SNX1 mutation. But in many cases, you'll see, for instance, there's a lack of immune response in SNX1, sort of an immune protection regardless of age. However, we're hitting some of those other pathways. Vesicle organization, protein folding actually makes sense, a lot of sense for the gene SNX1 and what its expected role is here. Okay, so we do this over and over. We can make a lot of mice. We can phenotype some decent proportion of those mice. And so we end up with massive charts like this, where we're looking at many, many factors across these 30 modules. Again, the same modules here, I labeled in a slightly different way, but they're in the same order. And then here, each of those mouse factors aged out to 12 months. And again, blue dot is a correlation, red dot's an anti-correlation. You can sort of pick through here. For instance, if we just put in sex as a variable, male is the variant in terms of sex, normalizing the female, you can see that there's a little less baseline inflammation response in the males in an Alzheimer's direction. A little blue dot, a few blue dots there under neural system genes, suggesting that there's already a predisposition towards those sorts of signatures in the males. We can look at early onset transgenes like the 5X FAD. We've looked at a lot of early onset transgenes, and they tend to really drive a limited number of those immune modules. So, this is basically amyloidosis. There's been a lot of work, now single cell work on the cell activation, the microglial response. And we find over and over again, that's limited to a few of these modules. So we are representing a certain amount of the disease there, but it's fairly limited. And as you can see, as we go to the right, it's really not representing those modules that we do tend to hit when we look at the late onset variants. Okay. The last thing I'll note here is we're looking at the effects of diet. Those effects of diet are very genetics dependent. So there's an interaction we're seeing between a high-fat diet and a PLCG2 mutation, where that mutation by itself drives a few blue dots, but you put them on a high-fat diet and suddenly you're driving a lot more inflammation, a lot of extracellular matrix effects, et cetera. So that's telling us that there's an environmental factor there, and so we're trying to involve more of those environmental factors, which of course bring in a very high, a large scale when we're doubling the background conditions for all of these strains. So we're limited in what we can do there. We can also look at responses along these transcriptomes in terms of preclinical drug testing. So we've taken the 5X FAD, which is this transgenic model. We needed to get started in year 1 with this sort of thing. So we just took something off the shelf and we dosed with, in this case, varebecestat. Sorry, I don't have that on the slide, but varebecestat was a candidate treatment for Alzheimer's disease, which was eventually proved not to be effective. But we wanted to do, show as a proof of concept that we could start to think about this. So we took some 5X FAD mouse, gave them 3 doses as well as vehicle of the drug, both males and females, well-powered study. You can see we have, expression changes for ApoE has much larger expression in the 5X FAD mouse. Maybe a little bit of drug effect there in the males, but nothing really strong. However, we can do the same sort of analysis and ask across the board, how do we see the effects of dose aligning with Alzheimer's disease? The idea, of course, for effective drug, we would want to see a red dot. We would want to see a negative correlation with human Alzheimer's and the drug effect. And so we can play the same game. Instead of looking at strains or diet, we can ask what's the effect of a drug. And so we actually did 2 experiments with 5X FAD, levetiracetam and varebecestat. Neither of them have been a successful treatment for Alzheimer's, but it was a way to get this going. And also a way to ask what the relevant biology being perturbed by those drugs is, right? So it's the same question, is that drug effect being mediated through any of these disease-associated modules? You can see levetiracetam really doesn't modify much of anything, a couple of modules weekly. Varebecestat is addressing some of that strong immune response and reducing the inflammation in the 5X FAD mouse, but it's not doing anything to help your neuronal phenotype there, which is a loss of neuron signature. Okay, so maybe maybe connecting with some of the, aberrant molecular pathophysiology there, but not stuff that really matters, which is to say, doesn't lead to cognitive improvement. The other interesting thing is varebecestat is, quote unquote, correcting some of those consensus cluster E modules. 5xFAD doesn't perturb them to begin with, so it's really not changing much in the mouse, but it makes us wonder if we were to, dose an ABCA7 mouse with varebecestat, would we correct what we saw in its transcriptomes? So we're ramping up to do this. We actually have a portal such that you can nominate compounds to treat a certain mouse model. And so we're taking that. It's called the Stop AD portal. And it's actually on the AMP AD knowledge portal that I talked about earlier. We can also, look at things like sleep, and taking these transcriptomes, we can compare with, say, sleep signatures. So here we're comparing sleep-deprived gene expression versus, spontaneous sleepers in BLAC6 versus our mutations. And you can see that in ApoE4 mutation in an aged mouse of 24 months, the effects you see in the transcriptome correlate, not super strong, but correlate significantly with what you see in sleep deprivation, and same with ABCA7. Looking at a group of genes there that are modified through sleep deprivation are also being modified in the same direction, ABCA7. So a lot of connections epidemiologically between sleep and Alzheimer's disease risk. Perhaps we can use some of these mutants to, phenocopy that risk or to understand that risk, at the molecular level. Other omics, we're also looking at metabolomics. This is a collaboration with Duke University and Helmholtz in Munich. We've run a couple of mouse models so far, 5xFAD and an ApoE4-TREM2R47H double mutant, which we call HAT here. We took 6-month-old mice, looked at both serum and brains. So of course, serum metabolomics is very exciting as a potential clinical tool, potential biomarkers there, biomarker patterns there. And so we're interested in not only understanding how our models might be, used as not only validation, but maybe even discoveries for some of those patterns, but also how you would then translate that information into the brain, which we have access to in the model system, of course, at multiple ages. And basically what we're showing here is that you see a lot more serum effects from the ApoTrem model than 5xFAD, whereas in the brain, you see much stronger effects from 5xFAD than the other. Not too surprising, those brains are absolutely full of amyloid in the 5xFAD model, so you'd expect to see more effects there than in the serum, but it comes through in the data. As we dug into this data, moving away from the principal component representations, you can see a very strong glycerophospholipid signature. So here's across all of our data sets, both in 5x and humanized ApoE4-TREM2s, we're seeing both in brain and serum some effects from glycerophospholipids. We can summarize all these data. Here I'm just showing the ApoTrem effects because they're very interesting. Within the brain, we see an increase in glycerophospholipids, as you can see on the left, for the most part, versus BLAC6, especially BLAC6 females. Whereas in the serum, we're seeing a consistent decrease in this panel of glycerophospholipids. So this is a summary of about 25 different metabolites showing this opposite pattern between brain and serum. So we were interested in, okay, we saw this in the mouse. Do you actually see this in the human data? So we messed around a bit with the ADNI serum metabolomics and Alzheimer's study, saw the beginnings of some of these patterns in the few metabolites they had in that publication. We then looked at metabolomics in the ROSMAP samples, which I mentioned earlier in terms of transcriptomes. They've also done metabolomes in both serum and postmortem brains. And here we can see that if we partition their study by ApoE4 carriers, we can see that the mouse serum effects, due to being an ApoE4-TREM2 carrier, matches what you see in the human serum effects for an ApoE4 carrier. So we're interpreting this as if it's being caused by ApoE4 in the mouse. We need really to separate the TREM2 off there to make sure. But as a first pass, I think it's pretty compelling that we're seeing both correlated effects across these glycerophospholipids in the serum and the brain. Again, there's a negative correlation in the mouse between the serum and the brain. And so that actually sets us up to make a prediction that what you see in the serum in the human may reflect an opposite effect in the brain. Independently, the mouse brain and mouse serum align with humans, but if you just look in the human data, you don't see this. And so, the human data is only about 90 samples. It's underpowered. I think we would argue here that there is some biology that you're not powered to see in the human, that of course you are in the mouse because we have controlled experiments. And so now, we're working with the ROSMAP team and Matthias Arnold in particular, who's been working on the metabolomics, to see if we can, look at larger cohorts and see if that anti-correlation is actually translatable, because it's suggesting a certain direction we might want to think, which of course suggests a direction of interpretation again. What you see in the serum may not have directly reflect what you see in the brain, but it may tell you something that is happening in the brain, in this case, the opposite effect of the glycerolphospholipid profile. We've also done proteomics, pilot samples, the same samples I showed before we did in the brain. Here again, strong effects from the 5X FAD model. The interesting thing here, though, in that second principal component is the early onset 5X FAD and the late onset genetics going in opposite directions. And so, this isn't too shocking given what I showed in transcriptomics, that we tend to hit sort of different modules. That's showing up in the proteomics again. And this is some TMT proteomics done in collaboration with Nick Seyfried at Emory. Proteomics are a very nice readout, maybe a little closer to the, the endpoint biology, another step away from the transcriptome onto the functional proteome. And so this, of course, will be something we can think of as a preclinical tool again. And so we tried that with that 5X FAD amyloidogenic model dosed with verbessostat. I'm just going to show this one module here. So here's a module, so you can do that same sort of module analysis we did in transcriptomes. You can do it with proteomes. It was done in ROSMAP. This is a module that comes out. It's called module 42 from the original analysis. The publication's about to come out. And you can see that all of these proteins are higher in cases, AD cases, than controls. And in fact, they're higher in what they call asymptomatic AD, which is brains that had neuropathology but had not yet been diagnosed for Alzheimer's. So the idea is that these may be people who are on their way to a diagnosis due to their neuropathology, didn't quite make it, but you're seeing maybe an early stage signature, which is a difficult thing in proteomics. It's quite likely with many proteomics, you're seeing sort of an end stage correlation with disease or a result of disease. So they liked the Emory group liked module 42 as it is linked to the asymptomatic cases too. It's a set of proteins, as ApoE is in there, APP is in there. So it's a reasonable, actually extremely reasonable set of proteins as a marker for Alzheimer's disease. And so, this module and others, we can do this analysis of looking for changes in our mouse models versus changes in cases versus controls. So here you can see 5X FAD by itself in the first row. That module 42 on the far right is the strongest correlation. So that's, in terms of the proteome, that's the module 5X FAD is modeling best. It models some others okay, and actually it goes in the wrong direction in a couple other modules. As you dose with verbessostat, low, medium, and high doses versus vehicle, you can see that we address, as you get to high dose, you start to reverse that far right effect on module 42, also called Navajo-White 2 module in this data set, where that blue dot is starting to be countered by a red dot when you dose high. On the far left, some of those, sort of beneficial effects of being a 5X FAD, whatever they are, those are difficult modules to interpret functionally, gets reversed. And so, I think a take home here is that verbessostat is reversing some of this, but not a lot of it. And in fact, 5X FAD isn't modeling a lot of this proteome very well to begin with. And so as a proof of concept, I think it was, successful in showing we can do this. As a real model of Alzheimer's disease, I think it's showing the 5X FAD is rather limited, at least in terms of molecular profiles. A point I wanted to make, of course, is sort of the correlation analysis is correlative. That is to say, it's not paying attention to effect sizes, it's more of effect patterns. And so just to illustrate that, here is that module 42, that blue dot I showed in the upper right of that chart. These are the actual proteins. This is their change in humans on the X and the 5X FAD mouse on the Y-axis. Very strong correlation there. Proteins that are up in cases are up in the 5X FAD for that module. We see a negative correlation for the high dose of verbessostat. So not as strong a correlation as we see from the genetic factor, but it's consistent across the proteins. But I think it's useful to sort of plot them together. And so here I'm showing the blue line is the same thing on the left. It's 5X FAD mouse response. The red line is the 5X FAD dosed with verbessostat. And so it's a minor correction. We're going in the right direction. We're starting to flatten that correlation, but which would take us, so to speak, back to normal. But it's not getting very far, although it is consistent across almost every one of those proteins. That, the red dot is lower than the blue dot. So we are pushing in the right direction. Of course, this is an experimental dose at a certain age. Maybe earlier it would have been better. But just to sort of give you a clear view of where we are with this, I think it's working. We can measure these things. These are not the right drugs and maybe not the right models at the moment. Okay, so I'll conclude there with a summary. I think hopefully showed that genetics and genomics in combination can help point out the biology of common disease. There's gene candidates often can identify pathways and processes, but the liability from these case control studies, the genetic level, are always very small, difficult to interpret. So we can add layers of quantitative measures like these omics to help us understand and interpret. And to do this, model organisms are extremely useful. We can manipulate them genetically. We can access tissues at any time in their life. We can do this high capacity screening. And I think one thing that we really pushed is that if we're really thinking about disease models and translational relevance, as we have been here, matching those assays helps a lot because we can compare like to like. Finally, acknowledgements. This is a big collaborative effort. A lot of people in my lab and other labs at the Jackson Lab. Big group at Sage Bionetworks. They basically exist to help analyze and disseminate data to the community, and they've been doing it all over the Alzheimer's universe. We're one part of that. So they've been a great help and collaborative group. Indiana University, where I'm sitting right now, in fact, is our main partner in Model AD, which has in recent years expanded to involve the University of Pittsburgh as well. And then Emory and Duke have been helping us with the multiomics. Thanks very much for your attention, and I'd love to take any questions.
Thanks, Dr. Carter. I always feel like I need to applaud or else there's like an awkward silence. I really enjoyed the talk. We do have some questions in the chat that I will ask. In the meantime, does anybody want to unmute themselves and ask a question? Hearing none for the time being, I will I'll ask, sorry, let me resize my window. I'll ask David Ashbrook's questions. He has two quick questions. What genetic backgrounds are the new models being produced on? And is there a version of those correlation tables that include behavior, which is also related to Anna's question about whether there's behavioral phenotyping going on in any of the mutants in terms of memory measures and frailty sort of things.
Yeah, I'll start with a behavioral question. It's easier. So we're doing frailty on all these models, and that's part of this screen where I was showing the 4, 8, 12-month transcriptome data. There's frailty data for all of them. All that, the models we've created, those data are up on the AD Knowledge Portal. We're finishing up our paper summarizing that first round as well. We don't see strong frailty phenotypes in any of these models up to 12 months. The models with promising omics, ABCA7 I mentioned, and MTHFR, were aging out to 24 months. And doing not only frailty, I think our frailty measure is 28 different measures. And also, I think we're doing limaze as well. We're doing additional behavioral phenotyping. Not a lot. I mean, I will say we weren't encouraged to do a lot of behavior in this project by the NIA, who is funding the center. Their feeling was that there was a lot of potential for omics-based preclinical testing, given that the behavioral mouse stuff has not been super translatable. And there are a lot of reasons for that. Maybe it is still a good way to go, of course, but they wanted us to develop these other methods, so we focused on them. Having said that, Stacy Rizzo is our lead at University of Pittsburgh, is a behavioralist, and is interested in finding the right behavioral assays that are most disease relevant. And somebody's waving outside the window. And so she's been thinking hard about how we integrate the most translatable behavioral assays as well into what we're doing. And so playing with things like touchscreens with mice to really get it and memory testing. The genetic background is a great question. So we're sitting here at JAX. We know that there are many flavors of mouse. We studied a lot of these. Everything I showed is on a Black 6 background. And that's partially because that's where the community is, and that's the reference, and that's how people think. So when we talk about an ABCA7 variant, it's interpretable. It's not at all clear that's the best background. It's a very resilient to neurodegenerative disease background. Lots of mice are, so it's not too surprising. And so we have started to address this. We took a panel of collaborative cross mice. We selected them for the most variation in gene expression of GWAS candidate loci. So we're trying to find CC lines that might be most likely to vary in the relevant phenotypes. It's a rough way to do it, of course, but that's where we started. We phenotyped them. We've crossed them to a transgene just to see how they respond to amyloid. Most of them are more resilient than Black 6. Which surprised us. We thought we would see, a lot stronger effects in a negative direction in the CC backgrounds, but we're not seeing that at the moment. We're still looking into that. We see sort of differences in, instead of amyloid plaques distributed more randomly across the brain, we're seeing a little more concentration in some of the models, a little more amyloid angiopathy in some of the models, a little different microglial responses in those models. So there's interesting biology there. In terms of driving us towards a better Alzheimer's model, it actually doesn't look likely that those CC strains are, which again is surprising because they're not as optimized for laboratory health as Black 6 is. So one thing we've been doing on top of that is to start to dig into some of the diversity outbred data. So the outbred representation of the same complicated 8-way mouse cross. Alyssa Chesler, who I think most people here know, has been looking at a lot of brains for her addiction center. So we've grabbed that data, and we're actually looking at baseline differences in pathways associated with Alzheimer's disease that we can map to an allele, right? And so if we can map that to a naturally occurring mouse allele, first of all, it's an interesting question is what's the candidate gene? Is that mapping to a locus that we see in human association? But even not, that's an allele that potentially could be a good driver towards a more susceptible mouse. And so we're starting to think about those allele swaps and how we maybe take that information and start to put it into Black 6 instead of starting with these more complicated backgrounds that are harder to understand anyway. To say that we put the cast version of Pillera into our mouse is a lot more straightforward and interpretable and experimentally manageable. So that's the direction we're going there, trying to get more of that genetic diversity in, because of course the information there is probably extremely relevant. We just need to know how to use it.
So you do think that eventually this might lead to us developing better animal models based on a human-like disruption of the transcriptomics, or that just has not yet happened yet? Or I was a little confused about what you said.
Yeah. So right. Yeah. So this is a little, in terms of in general. Yeah, I think we will get to more susceptibility to begin with the mouse backgrounds themselves. In terms of introducing the human variants, we've seen that we're already hitting transcriptome signatures of Alzheimer's disease that were not seen in the early onset models to begin with. So, so we're already, and we can think about some of those models which are not real Alzheimer's disease yet, they're not showing neuron loss and cognitive decline, may still be useful preclinical models if you're targeting that pathway and you just want to see if you're getting pathway engagement and you're not disrupting too much else in the in vivo model. So that's sort of the short term. The longer term, of course, we want full-blown Alzheimer's disease in a mouse model. We're not there yet. Nobody's there yet, because even the amyloid models don't trigger, tau pathology and what we think of as the causal neurodegeneration in humans. And so, our first step sort of is to try to get some of that amyloid pathology without those early onset drivers. And we're slowly getting there. I think we're seeing in aged mice, when we introduce ApoE4, humanized A beta, we begin to see more accumulation of amyloid. It's interesting, the APP gene has these 3 differences between human and mouse that, sort of look harmless, but actually lead to maybe stronger agglomeration of amyloid and drive more plaque formation. So we put those in. It was easy enough. It was one CRISPR to do all three. And so that's in most of these models. And actually, we're pretty optimistic now that we've seen that if we add, throw ABCA7 on that background and see if we're driving more processes that are going to lead to aberrant proteostasis and maybe more amyloid generation. And so I think that's where we are, sort of right in the middle of the process.
We're going over a little bit, but if you'll, people obviously can leave if they want, but if you'll permit me another sort of follow-up question to that.
Sure.
How far away, or will it be possible as we start to generate more transcriptomic data from Alzheimer's mouse models in general, and various drug treatments, to just sort of understand how the gene modules and the genes within them are perturbed by genetic modifications and by drugs? Can we start to actually predict whether a drug is going to alter transcriptomic profiles in a therapeutic way? And like we're generating, drug effects in non-Alzheimer's models, obviously. Like, how often can we just check other drugs that might have these unintended off-target effects? Does that make sense?
Yeah. I mean, if you've got a brain transcriptome, I can make a prediction right now, right, as to which model goes in the opposite direction of that brain transcriptome. And that we haven't done yet because we haven't got our hands on a lot of these data. I mean, it's very hard to get drug treatment data. Most of it's done privately. But in cases where we can, it's something we can do. I showed the example of sleep perturbation. It's the same idea, just taking a different data set and seeing if the same genes are going in one direction or the other. The sort of forward prediction beyond that, though, is an interesting question. So I'm also part of what's called the Treat 80 Center, that is really trying to just go through the humanomics and find drug targets based upon a hypothesis you would generate in a data-driven way to say that, oh, this module is perturbing this set of genes. The best guess is that, these are the key targets in this pathway, and here are 5 targets, can we intervene with them? Like, those are the ones where if there was a targeting built around synaptic communication, for instance, then we would just look down our mouse models and say, which one of these models is hitting that pathway? Maybe a couple are. Those are your preclinical tools to see if you can engage that pathway with that drug if you target that pathway. And that, again, that can be done independent of full-blown Alzheimer's mouse model. It can be done with the molecular signature that you're trying to target, which is a valuable first step, I think, to start to use this idea of precision medicine or precision targeting to see if it's really reasonable to try to do in vivo. So we're integrating well with those Treat 80 centers to set up the most predictive, highest priority combinations of mouse matched with compound that we might have.
That's great and really exciting, and I think potentially has a lot of really important impact. All of the stuff that you're doing, but both in terms of techniques to approach it, and then also the therapeutic impact of your work. I really appreciate it. I think I got all the questions. Oh, Anna, did you want to ask your question, or I can ask it for you?
Yeah, that's okay,
ahead.
Paula. I had a question, Greg. What do you think's going on with the non-coding mutants, especially BIN1? That's really a conundrum, huh?
Yeah. So we've just made the non-coding variants. We haven't phenotyped them out to any age, right? So we made sure we got the mutant in there. The BIN1 variant we went after, the best guess is it's a microglial enhancer. So it's a cell type specific enhancer that comes from Chris Glass's data in cell lines. And so, our hypothesis is that it's mediating microglial response to pathology, and somehow linked in with TREM2 and BLCG2 and these other variants in these microglial activation pathways. BIN1's really hard though in that sense, because BIN1's expressed in a lot of brain cells. The classic interpretation is that's actually an oligodendrocytic risk variant. And, well, maybe both. That may be what's driving that super strong association is there are multiple variants affecting multiple bin1 functions. So we've started with the microglial variant, hypothesized to be a microglial variant. And so we're building sort of a set of experiments around validating that hypothesis that, this is interesting to me because we're actually being more specific in our hypotheses with non-coding variants than coding variants. Because, with coding variants, we often think, oh, it must be modifying the protein some way. It's predicted to have an effect. So let's see what it does. And in phenotype level. And with non-coding variants, we're trying to be more careful because they are so much more difficult to understand. And so we'll see what comes out of that. You ask me again in 6 months, we should have the data.
I look forward to hearing about it. That's for sure. It was a wonderful talk. Thank you very much. A real pleasure.
Before everyone goes, I plugged a paper for Dr. Palmer that does the sort of drug discovery for cocaine substance use disorder. I put it in the chat. He sent me a private message, but I said I would plug his paper. So let's end there. Thanks, Dr. Carter. I was floored by your talk, Trevor, honestly, and I really appreciate you giving the talk. I think it was awesome and really important work, and I learned a lot, and you explained everything in a way that I could understand, which is saying a lot. So I appreciate it, and thanks again. Everybody have a great day.
Thanks, everyone.
Bye-bye.